All right, guys. So we're starting a new unit. We are doing an overview of the structure of the cell. So this should be review from Gen Bio, and it should feel pretty familiar. And we're going to go really, really quick through it. I've also been told that you can uh, Snapchat my face with like the face filters, and I would highly recommend that. Miss Bloom has sent me far too many far too many like Snapchats of my own face via this. So try it if you really want to. So part one, structure of the cell. Cell theory, we know that there's three basic components to what we know about cells. They are the basic unit of life, living things are made up of cells, and they only come from pre-existing cells. So those are the three things that kind of shape what we know about the rest of cells. There are two types, prokaryotic cells without a nucleus and without membrane-bound organelles, and eukaryotic cells. They have a nucleus and they have membrane-bound organelles. They are larger, more evolved. They came later on in evolution, and something that we have to realize is that cells are limited by their size. So their complexity is directly related to how big or small they are. The smaller the cell is, normally, the less complex and the less space there is for organelles. When we talk about the relative size of cells, we kind of want to talk about how if we look at something that could be relative, a chicken egg, and you'll see that is on our graph. A frog egg looks like just like a tiny, tip of the pen cap or like an eraser. If we divide that by 100, if we divide that by 1,000, that's going to be the size of most plant and animal cells, a little bit smaller, most bacteria and prokaryotic cells. We can see them under a light microscope. You can't see their organelles, but they also require an electron microscope to look with a lot of detail. The surface area increases at a slower rate than the volume for a sphere or a shape, and we'll talk about what that means. The larger the cell, the longer it takes for materials to diffuse to the center of the cell. So keeping those two things in mind, that can really impact the function of the cell, right? If I'm a really big cell, materials are going to take longer to get to my nucleus, right? If I have more, if I increase in volume, my surface area is not going to increase as quickly. And we'll talk about what that means. If we look at the idea of surface area to volume, right? And when we think about this, we think about like mitochondria with all of their folds and the Golgi apparatus with all of their folds and things with lots of like convoluted edges, like what absorbs in your digestive tract. The more surface area, the more absorption and the more space for things to happen. But as we get bigger, it makes it more difficult to get all that surface area, if that makes sense. So if you look, if I have tons of very small units, I'm going to increase my surface area a lot. So if I'm made up of tons of tiny, tiny cells, I increase my surface area a lot. Whereas if I'm made up of one big cell or I have one functional cell, I don't have as much surface area. And so there's not as much functional space. This is why cells are so tiny, because I want that large amount of surface area. When we talk about the basic form of cells, we look at prokaryotes. They are usually plasmid shaped or capsule shaped. They have their nucleoid space where the genetic material is. They will only have ribosomes, and they basically just have this cell wall. They may have flagella so that they can move, because a lot of the time they are single-celled organisms, and so they need to move on their own. Again, a close-up version of just genetic material in the center, ribosomes, and then plasma membrane, cell wall, and capsule. Usually because these are single-celled organisms, they need that cell wall to protect themselves. Looking at the eukaryotic cells, there's two types, plant and animal. Your animal cells are things that don't have chloroplast, a central vacuole, cell walls, or what we call plasmodesmata. These have all of the different parts that we will end up talking about. We'll talk a lot more about the organelles. In our eukaryotic plant cell, we see our chloroplast, we see that rigid cell wall, we see something called a plasmodesmata, and we see this large vacuole that stores water, so it'll keep its rigid shape. They do not have lysosomes, centrioles, or flagella, although some plant sperm, and you'll see it at the bottom, like the ones that are dropped off in the water and they have to get to the ova of the plant. If you remember talking about plant reproduction, they do have flagella. These are a couple different cells, that came from someone's cheek with different stains. This would be your animal eukaryotic cell. It's very clear to see the nucleus in all of them, depending on what stain it has. All of the other organelles are very, very small in comparison. So we talk about the nucleus first, because that is clearly the most present. The nucleus holds all of your DNA. It's actually a double membrane. We call it the nuclear envelope. So it's a circle within a smaller circle. It contains chromatin, and then in the nucleolus, which is the very center, that's where your DNA is going to be. It does have pores. We call them pore complexes, and that allows mRNA to exit to reach my tRNA, which is going to make my proteins. The nucleolus is the dark staining object in the nucleus. It contains the DNA, which codes for the production of ribosomal RNA. So the DNA is concentrated inside of the nucleus. It does not leave very often because it can be easily damaged, and that would cause mutations. So we know that the nuclear envelope, which is, the nuclear envelope, which is the tissue that surrounds the nucleus, the nuclear envelope is connected to the endoplasmic reticulum, and there's a purpose for this. If we think about the purpose of the endoplasmic reticulum and the ribosomes that are located on the rough ER, its primary purpose is creating and packaging and shipping proteins and sending them to their respective places. And so it's easy for me to directly get my RNA that's come from my DNA to my ribosomes to create my proteins, and then the proteins get shipped using my endoplasmic reticulum. The smooth ER is continuous with the rough ER, so it branches off from that. It lacks ribosomes, and what we know now is that, or hopefully what we remember, is that ribosomes are making those proteins. Probably the rough ER is mostly where the packaging and the labeling, if we think about it as like sending them somewhere, whereas the smooth ER is more specifically for sending things elsewhere in the cell. The Golgi apparatus is also going to be a multi-layered organelle. The Golgi apparatus, after the ER takes those proteins and packages them and determines where they're going, the Golgi apparatus is actually where they are sent. The membrane of the Golgi apparatus basically bubbles off into little transport vesicles and sends them wherever they are headed. And this is what it looks like. It's actually a really cool organelle. Functions, a little bit of an extension, modifying proteins, manufacturing polysaccharides, hopefully remember making sugars, and contributing to the plasma membrane. So if the plasma membrane breaks or is weak, the very malleable part of the Golgi apparatus can basically patch up and become a new part of the plasma membrane. Again, it directs secretions to where they need to go. Lysosomes. Lysosomes are going to be packages that carry foreign objects where they need to go. Basically, lysosomes are created off of the Golgi apparatus, and then they head to certain vacuoles to either deliver food or to get rid of waste. Vesicles and vacuoles are the membrane-bound compartments for storage. Vacuoles are going to be in plants, and they're very large. They usually store water to maintain the volume of the cell. Contractile vacuoles, on the other hand, are going to contract, obviously. They are different, and we won't really talk about these much, so don't worry about knowing these. The endomembrane or cytomembrane systems. This is just a larger term to refer to smooth ER, rough ER, and Golgi apparatus. They are all made up of plasma membrane inside of the actual membrane. And so they can fuse with the plasma membrane like we're seeing in the bottom right corner. We also have things called peroxisomes and glycosomes. We will talk about these in class if you have questions about them, but I'm not going to cover them here. One of the most important organelles that we see in animal eukaryotic cells are your mitochondria. Your mitochondria are used to phosphorylate ADP to ATP. So what we know is that the energy that we actually use is in the bonds of the phosphate to ADP. And so ADP is reused. Every time it becomes ATP, it's broken apart, and then it's recycled in the mitochondria. The mitochondria is highly layered. It has lots of folds, talking about surface area. And so all that surface area allows for a lot of ADP to be brought back to ATP. They believe that the mitochondria is an example of how the very first multicellular organisms evolved. The mitochondria have its own circular DNA, similar to a prokaryote or a virus. It also has its own ribosomes, and it replicates like a prokaryote. So you know that if you work out a lot, you're actually growing mitochondria. It also does protein synthesis, and so they believe that this is an example of a single-celled organism that could have lived on its own that's been eaten through phagocytosis by another cell. And they believe that this was the first eukaryotic cell, that way back when we were starting the earth, the first prokaryotic cells underwent phagocytosis. They ate another one, and they became similar to mitochondria, and they probably evolved to have different functions. And that's where we get our other organelles from. That is the end of this set of slides. Please stay tuned for 2 more, just talking a little bit more about the organelles that you need to know for Wednesday.